Welcome to Virology Live. This is session number 16. Today we are going to talk about acute infections. Welcome everyone. And before I start, just in case I forget, those of you celebrating Thanksgiving, happy Thanksgiving. Have a great holiday. We'll be back next week. And the other thing I wanted to say is, you'll notice at the top of every session slide, there's a little quote. And most of these are taken from chapters in Principles of Virology, the textbook on which this course is based. When I first started teaching this course at Columbia University, 12 years ago, I based it on the course, on the textbook, because I had written the textbook with a number of other authors. It was based on principles, not viruses. And so it was relatively straightforward to build a course on it. And most of the sessions are built on chapters. And so I have the quotation from each chapter, which is meant to sort of reflect what goes on in the chapter. And so this one, something's happening, but you don't know what it is, do you, Mr. Jones, is perfect for acute infections. So what is an acute infection? So we like to categorize things, we meaning, well, people. Everyone likes to categorize things, and scientists are no different. So we categorize virus infections two general patterns. One of them is acute and one of them is persistent. And so they are defined here on this graph, on this chart, where we're looking at time versus virus production in the blue line, and then the red is period of disease. Remember, we always speak separately of infection versus disease. It's not the same thing. And I find that that's one of the biggest misconceptions that people have about viruses, is that every infection causes disease. But as you know from listening to this course, it's not the case. Okay, an acute infection is one which has a beginning and an end. I think that's the best way to put it, has a very circumscribed period. It doesn't have to be short, but it starts and ends. And so here is a typical acute infection where we have a period of virus production. Virus production peaks and then it goes away, and this may or may not be associated with disease. And that's it. Then you're either killed or you're immune. And you can be reinfected, but the reinfection will have a milder disease. And examples of this include rhinovirus infections, common colds, rotaviruses, which cause gastroenteritis. We'll talk about those today. And influenza virus, which we'll also talk about today. And these acute infections, all right, the subject of today, acute infections, contrasts from Monday, next Monday, we will talk about persistent infections. They last the lifetime of the host. And here on the bottom are three different categories of persistent infections with different names. They're all persistent infections. We're talking here the herpes viruses, where once you're infected, it's for life and you keep getting infected. You keep getting virus reproduction and maybe disease also over and over, and others. And we'll go into this on Monday. Today's, though, is about acute infections. So rapid onset of viral reproduction, as shown by that graph. A limited, but not necessarily short, course of disease. It doesn't have to be short. I don't think that's part of the definition. It's limited, though. It starts and it ends. And it's ended either by death of the host or by resolution of the infection. And the disease can be severe or not. Could be even asymptomatic. It doesn't matter. That's the point. During these acute infections, large numbers of virus particles are produced, and this is how they are transmitted to new hosts. And this is a different strategy from the persistent infection, as we'll see on Monday. And eventually immune clearance takes care of this infection. All right, so that is the definition of an acute infection. And what I want to do today is go through a number of model acute infections so that you understand the characteristics of an acute infection and how they're expressed in particular viruses. So here is the course of a typical acute infection. Again, the idea being these are rapid and self-limiting. They're self-limiting either by immune resolution or by the death, unfortunately, in some cases, of the host. And this is just a repetition of the previous graph with more detail, and here we have the immune system included. We have virus reproduction on the y-axis, and then duration of infection on the x-axis, and then initiation of infection here at time 0, and the time. All right, so we have initiation of infection, and then, of course, our innate defenses, intrinsic and innate defenses, are engaged. And they may, in fact, completely limit virus reproduction, but in some cases it gets by them, as you know. And this red line is the, say, viral load. At some point when the innate defenses cannot handle it, there's a threshold over which the adaptive response is induced, and so we have induction of the adaptive response. We talked about all of this before, so you should know what's going on in these two areas here. And then we have a peak of virus reproduction, and then the adaptive response really starts to mature well after the viral load starts to decline. If you look at curves of immune responses versus virus production, that's often the case. And so the limitation of the infection is, in many cases, a consequence of intrinsic and innate defenses, at least for that first infection. And then you have memory, of course, and the virus is cleared or controlled. So this is a typical course of an acute infection, rapid and self-limiting. Now, the key here is also, where does disease occur? And so it depends on the virus whether disease occurs at or near the peak of virus reproduction or slightly delayed in comparison. We looked at some examples of that earlier when we talked about patterns of infection. Now, as you know, we have talked about this, that there are many inapparent acute infections, or is asymptomatic, right? No symptoms, and these are nevertheless successful infections. The virus is reproducing in the host. So there are signs for sure, right? Remember, the signs are things that you can measure, but you don't necessarily feel. The things you feel are symptoms, and no symptoms are disease. And you know why asymptomatic infections occur is not fully understood, but most likely represents a very vigorous initial innate immune response. Nevertheless, in many cases, these asymptomatic infections, the individuals are producing sufficient numbers of virus particles to spread in the population. And can you believe early in this pandemic, people were doubting whether asymptomatic people could spread the virus. Now, there is a reason to doubt it, okay? Because for some viruses, you do not spread infection until you develop symptoms, like Ebola virus. When you're in incubation period, you are not transmissible. But for other viruses, it is. So it is a reasonable question, but very early on, the data were produced showing that asymptomatic people could transmit infection. And then the remainder of the individuals who questioned it after that were doing so for political purposes, which has no place in science whatsoever. Now, how do we know that there are inapparent acute infections? Well, we do serological surveys, and we talk to people and say, did you ever have these particular symptoms? And they say, no, I've felt great for the last year and had no problems. Now, that, of course, is an imprecise science, but nevertheless, if you talk to enough people, you realize that you have a fraction of the population that has antibodies against West Nile virus, but never had any symptoms of West Nile disease. Now, these viruses that cause these acute infections and a large fraction of asymptomatic infections, these are well-adapted human pathogens, in my opinion. For example, over 90% of poliovirus infections are inapparent. Only 10% or so have some kind of non-specific flu-like illness associated with them, and of course 1% paralysis. This is perfect, right? Because the virus is reproducing in the host and it's moving very effectively to new hosts. It spreads throughout the population, without really causing very much disease. And I think that's a very well-adapted pathogen. We don't know what it was like when it first spilled over into people from an animal reservoir. That would be a really, really good question to address, but we don't have that virus, so we can't answer the question. But presumably it was more virulent in those days. And we'll talk more about that when we talk about evolution. I like to contrast poliovirus with norovirus, which we'll see today, which it causes vomiting and diarrhea. It's a gastrointestinal virus, just like poliovirus, but it's so unsophisticated. It has to make people do these things, and it's not necessary for really good transmission, as polio instructs us. And as we know, for SARS-CoV-2, there are large fractions of inapparent infections. The initial data said 20%, perhaps even higher. It really depends on the particular outbreak in the population. Now, acute infections are big public health problems, almost more so than persistent infections, because with a persistent infection, you have a lot of time in which to diagnose and prescribe an antiviral, if in fact you have antivirals. And so acute virus infections result in millions of infections every year. Influenza viruses, noroviruses. Of course, we are in a pandemic with SARS-CoV-2. It would be interesting to see as this pandemic subsides and SARS-CoV-2 remains endemic, which it will in the human population, how many infections we have every year. So we'll take a look at the numbers for influenza viruses, and it may approximate that. And the real problem with an acute infection is by the time you feel ill, the infection is really almost over, and it's already spread to other people. So that's why it's a public health problem. That's why SARS-CoV-2 caused a pandemic, because no one had immunity to it, and it spread silently. So just looking at symptomatic cases did not tell you anything about who, in fact, was infected. All right, let's do the first quiz question. Now, I was told last time that I should not go to the website when I read the questions to you, so let's do that. The website's running in the background, so I'll read the quiz questions. Which of the following do acute infections and incubation periods have in common? You're going to have to remember what incubation period was. I hope you do that. A, the virus is not replicating. B, no symptoms are visible. C, immune defenses are engaged. D, the immune system does not respond. E, all of the above. Okay, just going to remember what an incubation period is. And while you're chewing on that, and the reason people said the website is poor resolution, okay? I couldn't see it. Okay, so let's do some questions. Wow. Let's see how far back we can go to get questions. Oh, Zivka, happy birthday. You and your virome and your microbiome as well has a birthday today. Congratulations. Okay, so let me just announce this on the live stream. So as a way to raise money for Microbe TV, because we need funds to support what I'm trying to do here, which is, among other things, to hire people to help me so I can do more. If you contribute $1,000 or more in 2021, and it's tax deductible in the U.S., I'll send you a free autographed copy of Principles of Virology, autographed by me. I'll put a little note on it for you. And if you're interested, send me an email to Vincent@microbe.tv. I'll tell you how to do that. I'd have to build a web page with this on it for sure, with a button, but I haven't done that yet. All right, let's see here. There were some questions. It's cool that we're all here taking this class and you guys are getting to know each other and talking. And this is a really unique opportunity because in class in Columbia, the students have study groups. They get to know each other as well. But here you guys are all over the world. I think that's pretty cool. Very cool. Okay. I just want to point out that Mr. Ozzycam has a new icon there. Isn't that cool? I like that very much. So I have my Twitter icon is a virus, which I've had from the start, from the 9 years. I went on Twitter at the very beginning because I thought it would be useful. Now I don't go much because I think it's a cesspool in many ways. I mean, you can find good things there, but I find in the beginning, I told scientists to go on it, and they didn't. But now they get their egos stroked by being on it, so they're in it in droves. And I've withdrawn. I just post little things now and then, but I have over 100,000 followers because I've been at it for 9 years. Anyway, so I have my virus icon on Twitter, and people always say, oh, you should have a picture so people can get to know you. And I'm like, it's not about me. It's about the virus and teaching about it, although I do have a picture of me on the YouTube channel. So, I'm making a little concession to that. Okay, Frank says, I was born in the 40s, received both types of polio vaccine. Is there a test that will show me if I ever had inapparent polio? No, there is not. One could devise it. Well, if you've gotten both vaccines, you have the same vaccines, which is reproduced in you, so that makes it impossible to tell. But if someone only got inactivated vaccine, you could look for antibodies against proteins that are not in the purified particles, but there's no commercial test to do that. But it's theoretically possible. What's the shortest incubation period? Oh, well, there's a table coming up. We'll see. Can we say the persistent infection can't be removed and the acute can be? Well, we actually have ways to remove some persistent infections, as you will see on Monday. So I would just say they last the lifetime of the host, because now we can intervene with some persistent infections and eliminate them. Not all of them, but with some of them for sure. Ian has a question about vaccination, which really should be next time. How do you motivate people with one child death in 400,000 people? That's very hard, and I think we should save it for the vaccine discussion. But that's why a lot of countries have resurgence of polio, because they eliminate it, and then they say, why do we have to vaccinate? So it's not easy because you have to make a really good case for it. Where would long COVID be? It's going to be in the end of this lecture. We'll talk about it, okay? Is there an evolutionary advantage for the virus not to be symptomatic? Perhaps. That's from our viewpoint. This is anthropomorphizing. If you are not sick, you go out and about in the community and you infect. So I would say yes, because SARS-CoV-2 has a lot of asymptomatic cases and it's transmitting very well in the community, whereas SARS-1 did not, and so it didn't transmit well. So I would say that experience supports that conclusion, yeah. Are persistent and chronic synonymous? Yeah, those are two words. I call it persistent, right? But you could call it chronic. Latent is a form of a persistent infection with specific properties we'll talk about on Monday. I think misinformation is the virus with the shortest incubation. Yeah, it's very good. Indeed it is. Do kids keep having inapparent measles post-vaccination? This is a very good question. I'm going to reserve the answer because I'm in the process of reading the literature on it, because I would like to know if you are simply exposed and you get a little immune boost or what. I'll come back to that. Hopefully by the vaccine lecture I'll have the answer. At what point is the infection deemed over? Well, that's a very good question. So in my graph, it's over when there's no more detectable infectious virus. The problem is now we have now learned that fragments of RNA can persist for a long period of time after an acute infection. We learned that with Ebola virus. And I think we'll cover that in a subsequent lecture. I don't know which one, but yeah, we always thought Ebola was a classic acute infection. It still is, but apparently the virus can hide in immune-privileged places. So what do we do? Apparently it's very rare. Do we change the whole definition? It is still largely an acute infection. But you know what? It's never 100%. There are always going to be a few cases that break the rule. Okay, so I would say at the end of infectious virus production, it's over. And with that, well, let's go to the quiz now. So now we have to go back, and I have to now go to the website. And I know you don't like that, but you can't see it, but I'll try and read it for you. So let's see what we did. So most of you, but not all of you, and this is not a good score, 64% answered C. The immune defenses are engaged. That's what acute infections and incubation periods have in common. Okay? The virus is not replicating. Nobody picked that. That's good. About a quarter of you said no symptoms are visible. So symptoms, yes, is true for an incubation period. That's the definition of an incubation period. But an acute infection can have symptoms. So it's not correct to say no symptoms are visible. The immune system does not respond is incorrect. And it's not all of the above. So there you go. It's a tricky question. The tricky ones are what make you learn, in my experience. So today's cast of characters: influenza virus, poliovirus, measles virus, norovirus, and West Nile virus. There is a madness to my selection of these viruses. There always is, folks. I don't pick things randomly. Not after teaching this course for 12 years. There is a reason. These are viruses of different organs and that are transmitted in different ways. So let's start with influenza, which is appropriate because here in the northern hemisphere we are in the influenza season. Actually starts in the late fall. There are 3 types of influenza: A, B, and C. And when you talk about influenza, you're probably mostly thinking about influenza A. But in fact, A and B both cause a similar disease, and we vaccinate against both A and B. C, mostly inapparent or mild upper tract illness. We do not vaccinate against influenza C. And only influenza A causes pandemics, global outbreaks, every 10, 20, 30, 40 years. The last one was in 2009, and there will be another one for sure. Mark, not just my word, but most people who understand, there will be another influenza pandemic. Where they come from, we're going to talk about that in evolution and emergence. And the hallmark of influenza A viruses, and B as well, but to a lesser extent, is antigenic variation. We've talked about this before. The virus can change on a yearly basis and make variants that displace previous variants because they're more fit as a consequence of antigenic evasion. Interesting that no one ever says that the new flu variant this year is more transmissible. That's interesting, isn't it? They're just more fit because of antigenic evasion. Now, influenza viruses are transmitted by droplets that are produced by coughing and sneezing and talking. This is a summary of the droplet story, which we talked about before. And remember the Gesundheit, that's a machine that was used to have college students with influenza speak into. It shows that these droplets contain virus. But you don't actually have to cough and sneeze to transmit. Just talking is enough. You can also spread infection by direct contact. If you are playing with your nose and you have influenza and you've got mucus on your fingers, and then you shake someone's hand, you can transmit the infection to them because they will touch their nose and eyes. People touch their eyes and nose all the time. Ian Lipkin has claimed to me that he doesn't shake people's hands anymore. He only does the fist bump stuff, and he has cut down on his respiratory infections. Apparently didn't prevent him from getting COVID last year, right? You did listen to his episode of TWIV where he was recovering from COVID and talked about it. I hope so. Okay, so that's how this virus is transmitted. It comes into the nose and mouth and reproduces initially in the epithelial cells of the upper respiratory tract. And if the infection is limited to the upper tract, you have a typical uncomplicated influenza, but the virus can spread down into the lungs and reproduce throughout the trachea. If it goes down into the alveoli, it can also reproduce there and cause a pneumonia, a viral pneumonia. Right? So it can cause a primary viral pneumonia. Now, receptors play a big role like any other virus, right? The virus has to bind a receptor to start infection. And you remember that for human influenza viruses, the receptors are alpha-2,6 linked sialic acids. And if you don't know what that means, I tell you to go back to the attachment and entry lecture and review it. But it's a certain way that sialic acid is a sugar is presented, whereas avian influenza virus strains bind to alpha-2,3 linked sialic acids. Now, why do I even mention this? Well, most of our respiratory tract has alpha-2,6 linked sialic acids. Look, the nasal mucosa, the paranasal sinuses, the pharynx, the larynx, and only when you get down into the trachea do you have a mix of alpha-2,6 and alpha-2,3. You get down into the bronchus and the bronchiole and the alveoli, then you have a mix of the two. So the point here is that it's very difficult to be infected with an avian influenza virus because the upper tract, which is the likely initiation point of infection, doesn't have the receptors. And so when people are infected with avian strains, they have somehow inhaled the virus very deeply into the lungs, or there's another possibility, they got infected in the eye because the cells of the eye and the cornea, the conjunctiva, and even the nasolacrimal duct, that's a duct. Yeah, we all have that. It brings tears into the nose. They have alpha-2,3 linked sialic acids. So you could, in theory, be infected with an avian virus there. It would drain into your nose, perhaps, and amplify it to high enough levels that it would then drain down into the lower parts of your respiratory tract. And this has been shown in people who work on poultry farms. There have been outbreaks of avian influenza, which begin in the eye because of this. But most of the time, it requires the virus to get down into the lung. And typically people working with poultry, in many countries where these avian infections begin, especially H5N1, the high pathogenic avian influenza virus, they're actually sleeping with the chickens in their homes, and they're inhaling chicken breath all night. Actually, in chickens, it's shed in the feces, so they're inhaling chicken poop all night. So uncomplicated influenza, to give you a sense of this acute infection, incubation period of 1 to 5 days, depending on the dose that you get, the immune status of the host, abrupt onset, headache, chills, dry cough. You can always pinpoint the hour of the day that you start to feel crappy, and then it turned out to be flu. Absolutely, you can pinpoint it. Then initially you get high fever, muscle aches, malaise, anorexia. These are all flu-like symptoms caused by your innate immune response. Peak of fever within a day, and then it starts to decline. There's no fever by day 6, but your respiratory systems continue to intensify. Your cough becomes productive. What does that mean? Mucus. When you cough, out comes mucus, which is good. You're pulling it up from your lower tract, and you can swallow it. That's the mechanism by which we destroy viruses and other things that are in mucus coming up from our lower lungs. We swallow it, and it gets digested. And so this is a very short infection, but cough and weakness can actually persist for a couple of weeks because the virus does a job on your respiratory tract. It really, between the virus and cytotoxic T cell, they destroy cells that take time to regenerate. So how do we diagnose influenza? Well, in the winter here in the U.S., my students, they go to student health services with fever, cough, or sore throat. So influenza-like illness, fever at least 100 degrees, cough, or sore throat. They will say, you have flu and maybe give you a prescription depending on when you had symptom onset for an antiviral. They don't even bother to test anymore. There are rapid lab tests for antigens, which aren't very accurate. And so many physician offices, you can do them right in the physician office. They can take some nasopharyngeal wash and just do it right there. But they often don't. It's winter, it's probably flu. Beyond that, laboratories can do it by PCR, by viral culture, by serology, which are obviously more involved and take longer. Here are two experiments that illustrate the acute nature of influenza. So this is a virus that we can challenge people with. We discussed last Friday, I believe, yes, heavy metal flu fighters on TWIV. You know, that title was made by Dixon. He doesn't often have titles, but that was his, where they challenged people with influenza. And so that's what was done here. These are volunteers who were inoculated at time 0 with influenza. You put it in the nose, pipette it in the nose, and then we're looking at days after inoculation at virus shedding and symptom score. So they ask you how you feel. You know, they give you a number of questions about fever, malaise, anorexia, blah, 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 and they give it a scale. So we have virus titers and symptom score. And you can see that within a day after inoculation, you have virus shedding already. I mean, it's, 100, whatever this scale is, it's 100-ish, but not huge, but it's still gone up, right? And then by day 2, it's peaked already. It's a very rapid onset, right? And the symptoms are just slightly lag behind. So there's a little bit of shedding before, and this is shedding because they do a nasal swab, say, and they measure probably viral. This is an old experiment. They probably used infectious virus here. So it's just shedding. And then the symptoms peak when the virus is already declining. And then the two decline rapidly, and by 8 or 9 days, you have very few symptoms and very little virus. So that's good. And here's another experiment where we've looked at a little more. Again, it's a challenge experiment. We're looking at virus. Again, within 2 days, virus is peaking. And you can see this is an old experiment because this is a hand-drawn graph. I don't know if any of you are old enough to remember the days when we drew our graphs. I had these fancy pencil things where you could draw straight lines. We used press-on letters. Yeah, this is definitely a hand-drawn. You had a stencil that you would use to trace the letters with a pen. Oh, boy. Anyway, virus peaking. And then we're also looking at interferon. Look at that. The interferon peaks kind of coincides with the decline in virus, right? And this is nasal wash antibody. The interferon titer is also in the wash. Yeah, there you go, which is a good place for it to be. Febrile illness is starting at about day 1 through 6 or so here. And then look at, this is serum neutralizing antibodies with the big dashes, and then the dash, small dashes, that's serum anti-neuraminidase antibody. And this is nasal wash neutralizing antibody with the dash dot. So look at where they're starting to increase. Day 7, 8, it's already the illness is resolved. This is a typical acute infection. This antibody response will lead to memory, which will help you later, but doesn't help you this first time. You're going to be infected. You're going to possibly have some symptoms. You could have severe symptoms, but in this case, the infection is cleared. Now, in the U.S. and most of the world, there's huge surveillance for influenza, the most surveillance of any infectious disease. This is data from the WHO and NREVSS collaborating labs. They're a global network of laboratories, which, so in the U.S., the CDC would coordinate that, and then WHO coordinates it worldwide, where they get cases, they get samples from people, and they ask what virus is there, and they do antigenic characterization so they can plan for the next year's vaccine. And so these are examples of these data. Number of isolates on the left Y, and the right Y is % positive. And then these are different years, 2004, 5, 5, 6, etc., and months, the weeks of the year. So week 50 is week 50. It's almost the year is over, and then we start a new year. So you can see to the end of one year and through January, February, March of the other year, we have a peak. This is a seasonal illness in the Northeast in particular. This is the U.S. summary of the data. It's seasonal in our temperate climate where we have a winter. And you can see they have stratified these isolates according to subtype, H3N2, H1N1, which is the hemagglutinin neuraminidase type, and influenza B. And you can see that varies from year to year. And we currently have H3N2 and H1N1 co-circulating as well as B. And some years one predominates. So here the blue H1N1 predominated, and here the H3N2 predominated. And of course, in the next year, 2009, we had our H1N1 pandemic, a different H1N1 from this one. But the point of this is that it's highly measured and it is seasonal. And you can find this at cdc.gov/flu weekly. It's a really good site with all kinds of data. So what's the burden of flu in the U.S.? For this, these numbers come from studying influenza for many, many years in the U.S. And so it's a range. Obviously, some years are worse than others. And some years, and these are documented illnesses, 9 million to 45 million. 45 million in the U.S. alone. And people don't even blink, right? 140,000 to 800,000 hospitalizations, between 12,000 and 61,000 deaths. That's a lot of deaths. That's why we have a vaccine. And I don't understand why people say, I'm not going to die. How the hell do you know you're not going to die? And young people, very healthy apparently, people can die of influenza. Take your chance if you want. It's up to you. I have gotten a flu vaccine every year for the last 40 years. Here is the estimated burden by season, so you can see how it breaks down from year to year. Deaths in yellow, hospitalizations in, I don't know what that is, brown, illnesses in gray. And you can see some years, 9 million. There's the low end of this range, 9 million illnesses. There's the high end, 45 million, 2018. And of course, this went way down in 2020 because of all the masking and distancing and shutdowns and so forth caused by the pandemic. But this will be back. We already have outbreaks now. So here are some numbers from the U.S., just to give you an example. This is the 2019-2020 season, right? So 2020 season overlapping with the beginning of the pandemic. So the flu season typically over by May. But as you can see here, let's see, this is week 12. That's 3 months in. It's January, February, end of March. It's basically ended here. All right. And so here, these are week numbers. So 2019, week 40. That's how you would read this, week 42, 44. And we have H1N1 in brown here, which is predominating in that season. And you can see the peak here. Let's call that the peak. That's like January, like week 5 or so, February. And then it goes down. Now, 2021 through November, May through November, here you can see 2021, week 21. That's summer month. Well, here this early cessation is most likely due to pandemic preparations, masking, and so forth. And then the 2021 season. Let's see, this is week 37. So that's about right. But the numbers are much less. Look, number of positive specimens in the hundreds, whereas here we're in the thousands. So it's still suppressed where we are now, November. These data are from November 13th, not too long ago. Another way that you can measure influenza is by this metric called pneumonia and influenza mortality. So if someone dies of influenza, it may not even be put on the death certificate, or it may be marked as pneumonia only, or it may be marked as pneumonia after influenza. It depends. It really depends. So it's hard to estimate the burden of deaths, but this is an estimate. And this is a graph of the actual data every year for pneumonia and influenza mortality. Again, you can see it's seasonal, the red. In some years, we have an excess, a lot of deaths, which we call excess. The two black lines are the high and low that we would predict based on historical trends. And in some seasons, we exceed it, and some seasons we do not. So that's another metric, again, by week. And then we also look by state, of course. Influenza-like illness, that's ILI, activity level high in the reds, moderate to green in some states. And so this one was from 2019-20 at the peak of the season, where you could see there was influenza in just about every state. Here is the current one. This little graph, this little diagram or map at the bottom right here is from this week. And you can see very low influenza activity throughout the U.S. Just a couple of states have moderate to high activity. And even Michigan doesn't really show up, even though there's an outbreak there. The numbers are low. And so this graph is a little deceiving, because even though it is going up, as you would expect, the numbers are very, very low. And this is a historical graph of different seasons over week, for the week 40 through week 1 of the next year. So this comprises the influenza season. And then every year is a different color. So you can see here, typically we have a peak of influenza-like illness. Okay, so this is disease that's reported. A peak, at the beginning of year, week 2, week 4 or so. And the extent of the percent of visits for influenza-like illness varies from season to season. So here, 2017-18 was particularly severe. And I think we saw that here in this graph, 2018, the mortality exceeded the epidemic threshold. In other years, it's lower. So this one here is this brown one, for example, is 2015-16. It's much lower. And this one, this gray one, is the 2009 pandemic, which started in a weird time, right? It started much earlier than usual, and was in a decline by the time we would normally have our seasonal influenza. And the reason for that, it was a brand new strain, and immunity, lack of population immunity, trumps seasonality always. You can have complications of pneumonia, of influenza, primary viral pneumonia, where the virus goes down into the lung, destroys the alveoli, and causes inability to exchange oxygen. You can have secondary bacterial pneumonia. You can recover from influenza, and then a few weeks later get bacterial pneumonia because you're compromised, your lung function is compromised, your defenses are compromised. And that can cause a lot of deaths. Generalized muscle pain. There can be cardiac involvement when people are working very hard to breathe. This can put stresses on the heart and lungs. And there's another complication called Reye's syndrome, which is associated with encephalopathy and liver damage. The virus is not reproducing in them. We don't know what causes it, most likely a cytokine imbalance. We have lots of ways to intervene. We have non-pharmaceutical approaches. We have antiviral drugs of all different sorts. Tamiflu and Relenza are neuraminidase inhibitors. We'll talk about those later when we talk about antivirals. The Tamiflu is a pill, and Relenza is an inhaled antiviral. We have amantadine, which is not really used that much because we have resistance to it. Then the newest one is Xofluza, baloxavir marboxil, which is an endonuclease inhibitor. And you remember the endonuclease. We talked about it in RNA synthesis. And then, of course, we have vaccines, and we will talk about those when we discuss vaccines. So I do have to shift the question, folks, so pardon me for going there. Which of the following is characteristic of uncomplicated influenza? Transmission may occur via respiratory droplets. Incubation period 1 to 5 days. Fever peaks within 24 hours. Coughing and weakness can last for 2 weeks, or all of the above. All right, a lot of questions here. Can there be multiple acute phases? That's a persistent infection when you have multiple acute phases, as you saw in the graph there, yeah. I think we need to differentiate between infection and disease. Well, I always do, right? Hotel Papa 100, that's my shtick for the past year when I've been talking to people about COVID. There's a big difference, and people don't understand that. So, yes, I agree, but I have always done that. I'm still amazed I can get answers from you. Why wouldn't you get answers? I'm here to answer questions. If I don't know, I tell you I don't know, and I try and find an answer. Saint of Virology. That's interesting. My parents were Catholic, so I guess I could be a saint. So yes, Mr. Ozzy Cam has a poliovirus virion, right? Which is, I have a bunch of them behind me. This is this one. This is a little 3D printed model of a polio virion. Oh, it's so cool. Where is it? You can get this at Shapeways, I think. It's $30. And it's actually got quite detail on it, and it's made of some clay-like material. Yeah, I should make a bunch and give these out for smaller donations, yeah. Okay, what else we have here? I just checked the UK Weekly. Respiratory disease, still low. Flu, declining. RSV, increasing. Human metapneumovirus. Yeah, children still have plenty of rhinoviruses. So the pandemic has thrown a wrench in, but it'll get back to normal. Yep. I met someone on my last flight who's working on another study about SARS-CoV-2 transmission on airplanes. Yeah, they're interesting studies. We talked about one on TWIV not too long ago. Yeah. All right, biowarfare question we'll leave to another. Does flu bind to glycoproteins? Yes. Peter, you have to go listen to the earlier lecture. If you're taking this course, you got to listen to everything. No, just kidding. Yeah, it binds to sialic acid bound to other sugars in a chain, and the sugars can be bound to any protein. It doesn't matter. I'm sorry to show these because if you may feel like I'm promoting myself, but I just want to thank you for being thankful. Earth's virology professor, which bothers a lot of virologists. So then I say to them, so you do what I do, and then you can call yourself Earth's virology professor. Yes, listen to the TWIV special, Conversation with a COVID-19 Patient. That was good. He was coughing away. I edited some of them out, but I left some of them in. Do the rapid flu tests fail due to false positives largely? If you have flu, they're pretty good at picking it up. The problem is if you have something else, they're not good at that. Do virologists and ID docs generally get along? I have been sensing a lack of respect from doctors and from some conversations heard on podcasts. So, in general, I respect most people. If they show disrespect for me, I just throw them off the channel. I don't say nasty things. I have been known to say that such and such a person is, I usually will say they don't know what they're talking about, which is kind of a strong thing to say, but they often don't. And so I have great respect for infectious disease doctors. I work with Daniel Griffin. He's a partner of mine with the incubator and with TWIV and so forth. The problem arises when they say things that aren't correct, and I don't like that. I don't like when you say things that are wrong because you can find the information. So don't say wrong things. So viruses don't transmit during the incubation period would be a wrong thing to say, but many people will say that. So I would say that for the most part, they have respect for each other. But of course, many doctors have never dealt with a virologist, So we have we have infectious disease docs and other docs who work at academic medical centers, so they're interacting with PhDs all the time. So they've developed a mutual respect. I was about to say lack of respect. But docs working out, even over in New Jersey, would never see a PhD. They don't know what they do. I think they have respect. So my doctors, they're not researchers. They have respect for what I do. So I think there's always exceptions, but for the most part, it's good. And yes, Ed, it gets worse during a pandemic where everyone becomes an expert, right? I mean, I'm a virologist, so my thing is to understand things during pandemics and also between pandemics. But then other people jump in, like cardiologists who supposedly know all about infectious diseases, but they actually don't. And there are certain cardiologists on Twitter who you should not listen to because they're wrong most of the time, and they just repeat what they hear. Doesn't influenza often have GI symptoms? It can. It would be a consequence of cytokine production, but it's not because the virus reproduces there. Could we have a multi-antigen cheap rapid test? You could. Companies have chosen not to do it. Why? So the answer is, there wouldn't be enough of a market. So you would buy it. But the reason most people would buy it would be, oh, I'm infected with this and I need to get this drug. And there are no drugs. There are few for flu, but that's it. And getting the drug and having the test, it's a chicken-egg situation. You don't have one until the other. Use the slide rule. I used to use the slide rule. I had a lot of trouble with the slide rule. How many flu pandemics do we know the type of? Well, 1918 is really the first. 1918, 1957, 1968, and 2009. Those are the ones we really know. Could it be B? We don't have any contemporary evidence for B. Why would they just stop? So unlikely, I think. But because before 1918, we don't have much data, I really can't say definitively, right? I used to use calculators as soon as they came out because I had trouble with the slide rule. But I do have an old slide rule. It's a great wooden thing. And now, of course, I used to spend hundreds of dollars on a calculator with all kinds of functions. And now you don't need them because your computer or your phone has them. And if you want to do something more complicated, you just type it into Google, right? What proportion of influenza deaths tend to occur in older people? Well, the influenza deaths are older and very young. I showed the graph on Monday. Okay, go back to the lecture and look at it. Very young and very old have the burden of influenza death. This is an interesting anecdote. Only 5 positive flu A samples. Never happened. Do coronaviruses that infect chickens or birds pose any risk? So I don't think they are a risk because we don't have any historical evidence of them spilling into human, whereas with the avian influenza viruses, there have been. And we'll talk about that in terms of evolution later on. It is often said that the flu vaccine is low efficacy. So what we're looking at is prevention of disease. There are very few studies of prevention of flu infection. And so, 60% efficacy at preventing overall severe disease, and that goes down with age. One of the problems with the flu vaccine is it's only 60-ish%, and it doesn't induce good memory. So older people in particular need to get revaccinated. But also the virus changes, and that's another problem, that the virus changes from year to year, so the vaccine is rendered less effective. And so we need to deal with all those issues, and people are working on them. Okay, I think you might have emailed me about it, right? Jan or Yan. I think it was a reasonable email. And I said, yeah, that could be what's happening. And it's easy for me to just show the questions that way. Why is influenza seasonal? Well, we talked about that in a previous session. I don't want to go into it again because we have only limited time. That was infection basics. If influenza results in lasting symptoms, they're not lasting. They just can happen after the infection is resolved, and with time they will go away. So look at this. A smart ID doc would love to have one newer. No, not one newer. I mean, I'm not that brilliant. I just have worked on one thing for a long time. And in my old days when I did experiments, I had good hands and things worked. But I often challenge Daniel Griffin, and he's fully respectful. He said, yeah, you're right or you're wrong. We have a good conversation. And if you have respect, it's fine. The problem is in these public forums, people get disrespectful and they have an agenda and they start insulting you, and that's just not the way it works. And if you're in front of a person in real life, you're way less likely to insult them. And then in a forum when nobody knows what you are, right, who you are. So this argument about, does this aerosol expert keep saying flu like SARS-2 is transmitted by far-reaching aerosols? It can be, of course, but it's not the majority of transmission. So she may be an aerosol expert, but she's not a virologist, and I think you need to take both things into consideration. I'm not an aerosol expert, but you need to interpret the virology, and I don't think it's all one or the other. Sure, the problem is the very fine aerosols simply don't have enough virus in them to initiate an infection. And so the only way they will transmit is if you're in a room and breathing them for maybe 20, 30 minutes or an hour of them being constantly exhaled by someone else. Then you might get enough virus to initiate an infection. That's not the normal way. That's all I have to say on that. Could flu virus particles be found in bodies dug up? Well, they have been found in 1918. People dug up, not particles, but pieces of RNA. And RNA is tough because it doesn't last a long time. That's the oldest I know. There's actually a 1,000-year-old RNA virus recovered from from animal poop or something like that. I wrote about it on my blog a long time ago. But it's hard to get RNA as opposed to DNA, which is far more stable. But I think we have the tech to do it. We should be looking at that more. I would really like people to look at in the 1800s for, say, flu in people to see what was going on. Yeah. Some questions about vaccines we'll talk about later. According to the Wikipedia article, the 1557 flu pandemic is the first to be reliably recorded. So right now, you got symptomatology, right? You don't know it's influenza. And we're not going to know unless we got samples, right? We had the same problem with smallpox. So just be careful. Take it with a grain of salt. I know many people who have received both COVID and flu on the same day, different arms, right? No problem. Jan, short for Janet, sorry. But, I have to be careful because if it were Jan, which it could be, then people would say you got it wrong. So Jan, thank you. I appreciate it. Is it possible to clear out our latent herpes simplex? No. Unlike love, herpes is forever. What the genome-wide association studies prove that some people have a better response. They might, depending on what they're looking for. Yeah, they might. And finally, there were two 1918 samples, one from permafrost and one from paraffin-embedded, formalin-fixed lung tissues from Army soldiers who had died, and the U.S. Army kept them, and they were stored in their archives, and the RNA was still extractable in them. So there are two different sources. All right, let's go back to slides. Questions, sorry. What's the answer here? All of the above. 98% of you. Good. I don't have to repeat any of them because you all got that. All of these are features of uncomplicated influenza. All right. Measles is next. Paramyxoviridae family, envelope virus with 2 glycoproteins, a hemagglutinin neuraminidase, which has the HA and NA in one protein, unlike flu, which has separate proteins, and the fusion protein, which catalyzes fusion of the viral membrane with the cell membrane right at the cell surface, right at the plasma membrane. No low pH needed. And this, one of the most contagious viruses. And I will give you the R naught, even though Amy thinks it's a fraud to put this number, because you can influence the R naught, obviously, by interventions and your behavior. If everyone stays home, the R naught is less than 1, right? So even if you determine it early in an outbreak, you could be wrong because there's already an outbreak and things are already happening, and the epidemiologists will get very mad at me. But it's true. It's true. But it is very contagious. Pathogenesis, there's one serotype, and if you get infection, it gives you lifelong protection against disease. It's transmitted by respiratory droplets or maybe, and aerosols probably in this case, because you can infect someone. When I lecture students, and there's some all the way in the back there reading the New York Times, 100 feet away. If I were infected, I could infect them because this does transmit in those tiny droplets. And there's clear evidence for that, as opposed to not so clear evidence for other viruses. And you are quite contagious a few days before you develop the rash, which is the first time people would know that you have measles. As a kid shows up at school with some rash, school nurse, go home, you have measles. But before that, they're actually shedding and transmitting. But the cool thing, at least the useful thing, and nearly everyone who's infected shows a sign of disease. Very few asymptomatic infections. So the main community spread in the pre-vaccine era was the days before you have the rash where you're shedding. You can imagine in school, that's a great way to transmit it. So here's how the transmission works. So we inhale the virus in respiratory aerosols. It reproduces in our respiratory mucosa, gets into the subepithelial tissues. All this should be second nature to you now. Get into the lymph nodes. It eventually establishes a viremia. Yes, a respiratory virus gets into the blood. From there, it can spread to multiple organs, including the skin, and that's where you get the rash. One in 1,000 kids goes into the brain and causes encephalitis. Now, how does it get back out? Because you transmit it. Actually, the first question we need to answer is, how does it get in? It actually doesn't reproduce in the respiratory epithelium. I may have just said that, but that was a mistake. It actually reproduces in immune cells that are throughout the respiratory epithelium. Those are shown by this little orange and yellow cell here. That's an immune cell. That's what gets infected. The immune cells then migrate into the subepithelial tissues, and eventually the virus gets in the blood. So the epithelium isn't initially infected. So how do we transmit it? Aha! This is a very cool discovery not too many years ago. So the virus makes its way back through the circulation to the basolateral surface of the epithelial cells, where there is a receptor for the virus. It's called Nectin-4. The virus does then enter epithelial cells, reproduce in them. They're released apically, and that's how you transmit. All right, so again, there's no apical receptor in the respiratory epithelium for measles virus. It gets in through infecting immune cells, but it gets out by infecting the basolateral domain where there is a receptor. And that's why it's transmitted largely by respiratory droplets. And here on the bottom is the timeline of the disease. We have the incubation period where you've taken the virus in, it has reproduced in the immune cells, you have spread, you actually already have secondary viremia after that primary viremia, and then you have a prodromal period, which is the period before the characteristic signs of the disease. So now we're talking about 8 days after infection. We still don't have any rash, but now we have fever, and you are shedding from 8 to 14 days. You have Koplik spots in your mouth, little white spots, which are syncytia caused by virus infection. But not everyone's going to look in your mouth. A doctor should know to look. A school nurse should know to look. So if the kid says, I feel lousy, I have a fever, she looks in your mouth, sees the white spots. You have measles, go home. Of course, nowadays that doesn't happen because it's so rare, few people can recognize it because most of the people in the U.S. are immunized. And then we have, starting about 2 weeks after infection, this is a long one, we have our rash, and we're continuing to shed during the rash period, and we make antibodies, not Andit-Toby. This has been here for a long time. Every year someone tells me I never fix it. Sorry, it's an antibody. The rash, and it can last for 3 weeks and then recovery. Okay, so that's the pathogenesis. Really interesting, really different from what we've talked about. And uncomplicated measles, there's a child with rash all over the body. High fever, respiratory symptoms, runny nose, cough, conjunctivitis can accompany Koplik spots in the mouth. There's a Koplik spot right there, these little white thingies. Yeah, they're not easy to see if you're not used to finding it, right? And rash from face to extremities, all over the body. That's uncomplicated measles, but we do get complications, and this is a big problem. Encephalitis, 1 in 1,000 kids. Virus gets in the brain, reproduces, and this is not good, folks, because they always have long-term cognitive and sometimes motor issues as a consequence of that. Bronchitis, pneumonia, ear infections are also complications, and you can die. 1 to 2 in 1,000 kids die, but in countries, as I said the other day, with poor nutrition, up to 30% of kids can die after measles. And we have a very long-term sequelae. We have subacute sclerosing panencephalitis, SSPE. This is a disease that arises many years after the initial infection has been cleared, which raises the question, is measles a persistent infection? And we'll see the answer to that on Monday. And then, of course, it immunosuppresses you, as we discussed previously, and that leads to secondary infections and by other pathogens that you can't deal with because you're immunosuppressed. If transiently you're still immunosuppressed, and in many other countries there are a lot of other pathogens circulating, and that actually kills most of the kids in third world countries. And as you heard yesterday or Monday, it erases your immune memory. I want to show this slide again because, as Peter Palese told me when I was his PhD student, it never hurts to repeat things, especially to medical students. Now, that's not an insult. He was actually right because they often don't pay attention. They're reading the paper. I taught a lot of medical students in my life, and I'm telling you, when people bought newspapers in the back rows, they were always reading it, and now they don't have newspapers. They have laptops, and they're always on there, and they're not listening. So repeat it a couple times. Maybe they'll get it. When you get measles, it erases your immune memory. And this is an experiment using VirScan where they put thousands of peptides on a chip and then flow serum from kids over it and see which peptides react with serum. And you can see the kids with measles have lower antibodies to the viral peptides in their serum. And this is caused when the virus infects B and T memory cells and plasma cells, which of course make the antibodies. Before we had a vaccine in the U.S., there were 3 to 4 million cases a year of measles, 400 to 500 deaths, 48,000 hospitalizations, 1,000 kids a year with chronic disability from encephalitis. Now I ask you, do you want to take a risk that your child is going to have a chronic disability from a vaccine that has no side effects, essentially, other than the usual pain at injection site, fever, soreness, etc. It baffles me. And this is because people listen to anti-vaxxers, which are spewing false information. This is why we vaccinate against measles. In the U.S., we stopped all endemic transmission by 2000 by the vaccine, which was introduced in the 60s. Measles, mumps, rubella, MMR. Unfortunately, in 1998, the Wakefield report out of the UK, which said that the vaccine caused autism, led to decreased immunization among parents who were confused, right? They didn't know what to make of this, so they didn't immunize their kids. Big outbreaks in the UK and Ireland, and those viruses were imported into the US, where we have had outbreaks periodically because certain groups of people choose not to be vaccinated. For example, Rockland County, New York, big outbreak a couple of years ago in a predominantly Hasidic community whose leaders said we shouldn't use this vaccine. He was misinformed. So here's the effect of vaccination on measles. Lots of cases before the 60s and almost eliminated in the U.S., but as I said, because of vaccine hesitancy, we continue to have outbreaks, and that's shown here. Measles by year in the U.S., some years very low, other years we have big outbreaks. Here in 2014, I think this is one associated with Disneyland in California. You know, a whole bunch of parents who didn't vaccinate their kids all went to Disneyland. Here's another one in 2019. And very few cases in 2020 and 21 because of the pandemic and so forth. If you look globally, there are a lot more cases because in many places we have not vaccinated sufficiently. And this is from 2016 through 2020. For example, in 2019 alone, globally, 400,000 cases plus of measles, and a good fraction of those fatal or encephalitis. And these are colored by WHO region, right? The African, the Americas, Eastern Mediterranean, European, Southeast Asia, Western Pacific. Those are the WHO regions. They divide the world into those, and you can see the greatest burden in the African region where we don't vaccinate sufficiently. And here are the top, these are the countries globally with most measles cases. The dark colors are the countries with the most measles cases. You see parts of South America, Africa, and Asia. The top countries in, I guess the last year, Yemen, Tanzania, India, Nigeria, Pakistan, Burundi, China, Brazil, DRC, and Kenya. They had the most cases. And many countries put off their measles campaigns because of the pandemic, so I assume these numbers are going to go up in big time in the next couple of years. All right, next quiz. All right, back to that. Which of the following is a good reason to get measles vaccine? A, there's a 1 in 1,000 chance of acute post-infection encephalitis, 1 to 2 in 1,000 chance of death. Each infected person spreads virus to 15 others. Immunosuppression can lead to secondary infections. Or all of the above. Okay. A lot of people talking about their measles experience. So measles has to go all the way via blood and back. Yes. You don't apply human logic to it. Okay, that's wrong. That's anthropomorphizing. Because we think it's like to get to New Jersey, I go to Long Island and back, right? Viruses are just evolving, and this works. It works for them. Is it not so if measles is not replicated in the ciliary cell, does that mean it will leave the cell and it will be back for the shedding? Yeah. So remember, it comes back via the circulation because it's in the blood. And some of those capillaries go under the mucosal sheet, and the virus is getting out and infecting at the basolateral domain. It's a little counterintuitive, but that's what happens. Yeah, so this is measles. German measles was the chicken pox, right? That was the word for chicken pox. All right, so I'll bring this up because, Jenny, it's suspected that herpes plays a role in Alzheimer's. It's being studied. So for you to say the pharma refuses to study, the link is simply wrong. I can tell you a number of clinical studies just at Columbia where patients are on anti-herpes antivirals for 3 years. It takes 3 years to do the study. So don't say that Big Pharma is not in on it, because they are. If it turns out that acyclovir prevents it, they're going to sell more acyclovir. So if you don't know what's going on, you shouldn't criticize it. And I don't mean to get upset with you, but because I know you often come to the live streams and so forth, but it's not correct to say that they're ignoring it. It is being done. Just look it up in clinicaltrials.gov. You'll see the trials that are going on. Now, I don't know if acyclovir is going to help, but they're looking at it. There shouldn't be any measles. That's right. There should be no measles. Yes, so measles, by the way, can be eradicated because it's only in people. And in fact, it's on WHO target for eradication next after polio. We could get rid of it, just like we got rid of smallpox. How is measles able to remain viable and infectious outside a host for so long? It doesn't remain viable outside a host. There's always human-to-human transmission. And there are places in the world where there's a lot of measles, as you saw in the graphs, and it's easy for that to travel elsewhere. It's not that the virus is surviving in the environment. There are people carrying it all the time. And this is why influenza virus returns every year, because it's always present all year round. In New York in the summer, there's influenza. Absolutely. Just at a very low level. But Mr. Ozzycam, you can argue with the quiz, but it's right. Okay? So if you got it wrong because of that, you're wrong. On average, yeah, but I would not make a question wrong because I left out on average. You're getting a little pedantic there. Which I shouldn't criticize because I'm always right. German measles is rubella. Okay, sorry about that. You're probably right. Yeah, it's rubella. Varicella is chickenpox. My bad. Yep, varicella. Okay. I knew I was going to get that wrong when I said it. Let me go back here. Okay, thank you. We don't need 10,000 people to say it. How can virus erase immune memory? It infects memory B cells. Yeah, apparently it infects it not. It doesn't erase it. It decreases it substantially. Okay, so it gets most of the cells. Yeah, it's viremic. It can get everywhere. T cells are not affected by measles, the loss of immunity not be so bad. Well, it depends on the timing, right? You can restore your B cell immunity by being reinfected or revaccinated. But if you don't do it in time and you get infected, you could die, right? And that's why a lot of these kids in underdeveloped countries die, because they get opportunistic infection by immunosuppression and loss of immune memory. Yeah. Okay, that's enough here. We're getting into weird things in the chat. Let's see what happened here. These are all good reasons. Every one of them is a good reason to get vaccinated. Encephalitis, 1 in 1,000 chance, 1 in 1,000 chance of dying, spreading it to 15 other people, and leading to secondary infections. What's next? Polio. Here we go. Polio virus, which is right here on my desk. My favorite virus. Why? Because I worked on it for 40 years. When you work on something for 40 years, no matter how evil it can be, and polio can be evil, right? Becomes your favorite. An RNA virus made of an icosahedral capsid with a plus-stranded RNA genome inside of it. Pathogenesis is as follows. You ingest something that's contaminated with poop from a person who is excreting poliovirus in their feces. You ingest it, the virus passes through your stomach into your intestines. It survives the low pH and enzymes in the stomach. It survives the high pH in the small intestine, and then gets into your intestine and starts to reproduce in the cells lining the intestines, gets into the blood via the lymph, and in 99% of people, no paralysis develops. But in a certain fraction, the virus seems to get into the spinal cord and brain, 1% of infected people. We think that from our work on transgenic mice that the virus gets into the nerve endings in muscle. So it comes out of the blood. The nerve endings have poliovirus receptors. The virus is internalized into the nerve. It's then transported on the microtubules by motors. This would be dynein motors bringing it towards the cell body, and then can reproduce in the neuron cell body, and it can also spread up to the brain. And if you destroy enough of these neurons, then you get paralysis. And so here's a slide from my laboratory many years ago. It's a section of spinal cord from a transgenic poliovirus receptor mouse where we've inoculated these mice with poliovirus. They became paralyzed, and we took this section. And these green dots are viral RNA, and those are neurons. So the virus is reproducing exclusively in neurons within the spinal cord, and that's because those are the cells that have the poliovirus receptor on them. And look at this neuron here with its axons and dendrites, just full of poliovirus RNA. The green dots are poliovirus RNA. And you also see lots of immune cells coming into the cord to try and clear infection. Eventually, you get enough neurons destroyed that innervate a particular muscle, and you get the paralysis characteristic of the disease. So here's another hand-drawn figure. This is from a 1950s textbook of medicine showing the pathogenesis of poliovirus. We have exposure here and then days after infection. And we're looking at different phases of infection, different parameters. So at the top here, we're looking at temperature, right? So after about a 7-day incubation period, you get a little spike in temperature, and then it goes down. And at that time, you already have virus in the feces. You have virus in throat secretions because it's reproducing there as well. You have a peak of virus in the blood, right? And then in some people, the virus reproduction continues. You can have paralysis. You have a huge spike in temperature at that point, and you have virus now in the central nervous system. That's here at the bottom. This is virus in the central nervous system and virus in the blood here on top. Big spike. So you see earlier than about 10 days, no virus in the CNS, but in those 1% of the people, it gets into the CNS, you get paralysis. And look at the virus in the feces, how long it persists for 30 days. And here they're looking at infectious virus. They didn't have PCR in the 50s. So this is infectious virus. So you can be shedding virus a long time, whether you're paralyzed or not, of course. And 99% of the people are not paralyzed. They're walking around shedding virus, and that's why quarantine doesn't work. So we are the only... I'm so sorry I didn't share the slides. Damn it. How far back do I have to go? Yeah, I'll go back to polio. All right, so you can remember what poliovirus is. The pathogenesis, it's ingested, reproduces in the intestine, spreads to the blood. 99% of people are okay. 1% the virus invades the central nervous system. We think the virus enters the nerve endings in the muscle via poliovirus receptors, transported by dynein motors to the cell body, reproduces in the cell body, and then spreads to the brain as well. So if you destroy enough neurons in the spinal cord and brain, then you will get paralysis. And here's a section of a spinal cord from a transgenic poliovirus receptor mouse. This was done in my lab years ago. We infected the mouse with poliovirus. It became paralyzed, and we took these sections. And in the cord, all these green dots, we have hybridized this section with an RNA probe. So this is showing viral RNA. And these are just neurons here where the virus is reproducing. No other cell type, because only the neurons have the polio receptor. Here's a neuron on the right. You can see the cell body, the axons and dendrites. Those are the projections going away and towards the nerve, and they're full of RNA. Look at how much RNA is in it. And there are also immune cells coming into this infected spinal cord. They're, of course, trying to clear the infection, and what role they play in the neuronal destruction is a good question. We really don't know. So this is the wonderful picture I already described, where from a 1950s textbook of medicine, we see hand-drawn different phases of infection. So these are observations from cases of polio, also some non-human primate infections, but no challenge experiments could be done with this virus. And we have exposure and then days after infection. So we can see here at the top the temperature. After about a 7-day incubation period, we have a spike of temperature. And most people then get well after that. You may have some headache, nausea, sore throat, flu-like illness. But you already have a lot of virus in the feces and virus in throat secretions. You have virus in the blood. And then in a small number of people, the 1% of the infection continues. There's now a second peak of temperature after 14 days or so. That's accompanied by paralysis and virus in the central nervous system. So 1% of individuals, the virus gets into the CNS. But in all cases, you can see the virus in the feces goes for a very long time. And this is actual infectious virus here, because they didn't have PCR in the 50s. So that's infectious virus. So you can transmit, whether you're paralyzed or not, you could transmit for a long period after infection. So we are the only known reservoir, and this is why polio is eradicable. It's transmitted pretty much exclusively by fecal-oral transmission. There's some sense that in certain populations there might be a saliva component, but not clear. It is seasonal. It peaks in the warm months, in temperate climates. And there is a long-term sequelae called post-polio syndrome, which happens 30 to 40 years after you had your first bout of polio. You suddenly get paralysis again. So many people could be given physical therapy and regain their limb function, have normal lives, but then 30 to 40 years later, suddenly they're paralyzed again. They end up back in a wheelchair. Why is that? It's about a quarter to 40% of all cases. So we still have people today developing post-polio. The last cases of polio in the U.S. were in the 70s. There's no virus left in those patients. It's not an infectious process. It's not like long COVID, I think, where the symptoms continue. This is more like a post-acute sequelae, you're fine, and then suddenly you get paralyzed. Why is this? Well, it's not virus. There's no infectious virus left. What happens, we think, is that as you age, you normally lose neurons, but you have enough so that it doesn't impact your motor movement. If you've had polio, you've lost a lot of neurons. And as you age, the loss of additional ones now, it makes it impossible to use some muscles. And so that's what we think is the reason for that. In the U.S., we used to have many, many cases of polio peaking in the 50s. The baby boomers, post-war baby boomers, of course, amplified those numbers. 1955, an inactivated vaccine introduced. 1962, the oral replication competent vaccine. Polio eliminated in the U.S. in 1979. And we'll talk more about this when we talk about vaccines. Now, for today's factoid, did you know that in any 24-hour period, about 200 million people on the planet have gastroenteritis, and the amount of diarrheal water they pass is equal to the volume of water passing over Victoria Falls in 1 minute, which is 65 million liters per minute. That's a lot of diarrhea. And only a gastroenterologist could have told me that. I got this from Harry Greenberg, who is a Brooklynite who is a gastroenterologist, actually a virologist, basic scientist, but also a gastroenterologist at Stanford. Wonderful guy. He gave me this slide. And most of this is caused by norovirus, a virus we haven't talked about much in this course. It is a plus-strand RNA virus, non-enveloped, has an icosahedral capsid. The family is Caliciviridae, responsible for 50% of all foodborne outbreaks of gastroenteritis in the U.S. 48 million a year of people eating certain kind of food and getting gastroenteritis for a couple of days. And here it is, the pie chart of all these foodborne illnesses, 58% norovirus and then a bunch of other bacteria. We didn't always know this. We thought it was all bacteria. And then a number of years ago, norovirus was identified in these patients, and this is the big agent. And this is a virus that we can do challenge studies with. We can challenge humans because why? Well, you just throw up and have diarrhea for 2 days. It's not fatal, as long as they give you fluids. And so you know what they do in medical schools on Friday? So we couldn't grow this virus for years. So what they would do, they would take people with norovirus diarrhea, take the diarrhea, filter it to sterilize it, which turned out not to be a great thing, but we'll talk about that in a moment. Then they put a flavoring in it, and then they have people drink it. So the medical students on Friday would drink the norovirus, and on Saturday and Sunday they'd be throwing up and having diarrhea. But Monday morning they're good to go back to class. And over the weekend, of course, you can do measurements on them, and they get paid $300 to do that. So we can do challenges. And it turns out that you only need 18 viral particles to infect you. That's one of the few viruses where we know. I can tell you 18 SARS-CoV-2 particles will not infect you. The experiment hasn't been done, but judging from infectivity studies, that'd be my guess. Okay. Norovirus is what I call a two-bucket disease. What is a two-bucket disease? I think you can think that, you need a bucket to vomit in and a bucket to have diarrhea in. I got that from microbiologists, but I like it, a two-bucket disease. It's fecal-oral spread. The particles you ingest in food, they pass through your stomach. They retain infectivity, very much like poliovirus, and they seem to reproduce in the epithelial cells. You can see the villi in the proximal jejunum. Here is normal villi. Here's villi in a norovirus-infected person. So you could take biopsies from these volunteers and so forth. And we don't know why it causes vomiting and diarrhea. We have a sense for rotavirus, right, because it makes a toxin that causes that, but we don't know why it happens with norovirus. And it turns out for years we could not grow norovirus in cells in the laboratory. It turns out you need bacteria to enable them to grow. And so normally what scientists would do would be to take diarrhea from patients who had norovirus gastroenteritis, and they would put it on cells in culture, and but never could grow it. And what they always did was to filter it first to remove the bacteria, right? Because the bacteria will contaminate your cell cultures. Well, one day someone forgot to filter it, and it turned out that now the norovirus could grow. So here's an experiment where we're looking at cells in culture. These are B cells, actually, because it turns out that B cells in the gut are one of the targets of norovirus reproduction. So we're looking at genomes per well. This is by PCR with time after the infection of the B cells in culture. So if you filter the diarrhea to remove the bacteria, it doesn't reproduce. But if you don't filter it, it reproduces. So some reason bacteria are needed. The idea is in the gut, the bacteria facilitate infection of the epithelial cells. So we believe that the noroviruses infect intestinal epithelial cells, the ones that are called IECs here. Those are the gray ones. And also macrophages, dendritic cells. So the macrophages are shown here, the green guys. So the virus can get into the subepithelial tissues and infect macrophages. Dendritic cells, those are the orange ones. T cells with the big blue nucleus, and in particular in immunocompromised hosts. And that's where the infection can be the worst if you're immunocompromised. But otherwise, you just keep fluids high and you'll be okay. And B cells. So the B cells are in vitro. We're not sure what the relevance of that is to in vivo. But the bacteria are somehow facilitating infection, perhaps by allowing the virus to be taken up into cells. Not quite clear what's going on. But this reproduction in these cells, the blunting of the villi, causes the diarrhea and the vomiting. And, of course, the virus is spewed out in the diarrhea and infects other people. So it's transmitted fecal-orally, also by aerosols. If you vomit, you generate an aerosol, and you can expel virus as well. Contact with contaminated objects, fomites, food, water, so environmental contamination. If there is sewage in water that is used for food preparation, that could do it. The foods can be contaminated at the source, so oysters will take up a virus from the waters if the waters are contaminated. Raspberries if you wash them with contaminated water. Or you have food handlers who are infected but don't have symptoms. They're working. They don't have good hand hygiene. They will contaminate the food as well. Short incubation period, sudden onset of vomiting and diarrhea and stomach pain. All of a sudden you start vomiting and diarrhea, 28 to 60-hour illness. And this is here the key, 30% asymptomatic. You get infected, you don't get vomiting and diarrhea, so you go to work. If you're a food handler, you contaminate the food. And that's why there are the signs in the restrooms everywhere because of norovirus. Every restroom in a restaurant you go into, at least here in the U.S., employees must wash their hands, and that's because of norovirus. Virus shedding peaks a few days after illness onset, but can go on for up to 2 months. And so you can get better and go back to work and contaminate people. And immunity doesn't seem to be so durable. Short term, you get reinfected later in life, probably because mucosal immunity is low, and that's what you would need to protect you. But the reinfections can be milder. We are the reservoir of noroviruses, but animals can certainly be infected, and we're worried that maybe sort of like SARS-CoV-2, we're establishing a reservoir in non-human animals. Everybody's affected. These occur year-round, although they do tend to peak in cold weather, and the outbreaks occur in semi-closed environments, nursing homes, hospitals, and cruise ships. The military has outbreaks, schools, anything where a lot of people are together, that's where you get norovirus outbreaks. Nothing we can do right now except give you fluids to prevent dehydration. There are vaccines in development. And, in kids, 200,000 deaths a year because they're not properly hydrated, especially in countries that can't hydrate kids with either water or intravenous fluids, electrolytes, and so forth. So here's the breakdown of transmission, and according to settings on the left. So you see the biggest percentage, restaurants and catered meals. Then we have retirement centers, nursing homes, hospitals. Cruise ships is actually less than 10%, but they get a lot of publicity because they have to come back from their dream trip and everybody has to leave and they have to decontaminate the ship, and it's really very expensive. Modes of transmission, 88% person to person, where there's a food handler involved, and only 10% foodborne. And this happens on airplanes. Here's an article from an Air New Zealand flight where somebody was vomiting into the bag, and the crew cleaned up the mess, and then on every successive flight, at least one or more crew members got sick. So, vomiting will make the aerosol, and then cleaning up the vomit, of course. So it is quite infectious. And as I said, that these outbreaks occur on cruise ships. This is just a listing of various cruise ships in 2010, sailing dates, where there have been outbreaks on the ship causing them to come back. Sometimes they're norovirus. Sometimes there are other viruses associated with gastroenteritis. We know of other viruses, but they don't always figure out what they are. There's a certain cruise ship line that calls their cruises the happiest place on earth. Personally, I would never go on a cruise. Be in that tin can with thousands of other people and eating meals prepared in the same, no, just not happening. Now, I used to have a cruise ship person. I don't remember what his position was. He was from Norway, and he used to do things with cruise ships. And he said, we're getting better. They actually have these automatic hand washers on the cruise ships, he said, where you put your hands in and you get automatically washed and dried, so there's no excuse for no hand hygiene. But I'm still not going on a cruise ship. But why are they associated with cruise ship? Well, we track the infection, so we find them very quickly, faster than we would on land. Close living quarters, right? And new people also, passengers can bring them into the ships as well. So how do you protect yourself? Wash your hands often. Hey, does that sound familiar? We were talking about that before, noroviruses. Fruits and vegetables, you better rinse them really well because they might have been, don't ever trust a package that says pre-washed. It's not good enough. Rinse everything. Cook your shellfish. I know you like to eat raw oysters, but man, it's taking a risk. Well, if you don't mind 2 days of vomiting and diarrhea, then go ahead. Clean your surfaces well. If you're sick, don't prepare food. And folks, don't eat the mints when you leave the restaurant because studies have shown they're contaminated with fecal bacteria because people go to the bathroom before they leave. They don't wash their hands and they take a mint. Even if they're wrapped, the wrapping is contaminated. Don't do it. Okay. Last virus. Oh, not really, but we're going to talk a little bit about COVID. West Nile. And this cartoon shows you what I'd like to say, that this has not come from Egypt. Although this cartoon implies that it does. It did. It was isolated in 1937 in the West Nile district of Uganda, not Egypt. The West Nile district. But we did not have it until 1999 when it came to New York first, here in the summer of 99. You should read Dixon's book, West Nile Story. Well, yeah, you should be able to get it on Amazon. He tells the story of the first year or so. Very interesting story. Apparently came from Israel because the New York isolate sequence is identical to that of an isolate from a goose in Israel with no changes. So it somehow got here from Israel. We don't know how. Infects many birds, many kinds of mosquitoes, many vertebrates in nature. And these are the countries in green where we see now extensive West Nile virus. This is a virus of birds and non-human mammals. and it is a flavivirus transmitted among them by mosquitoes, primarily culisine mosquitoes. Humans and horses are accidental hosts. When we get infected with West Nile virus, it's a spillover from a bird or some rodent through a mosquito. We do not transmit it to other humans by mosquitoes. We don't have high enough viremia to do that. So every West Nile case is a spillover from a bird or a rodent or something. And same thing with horses. And in horses, this can be serious. It can kill them. So there is a vaccine for horses because racehorses are expensive. And even the horse you may buy and you love, they're not cheap. And so you got to protect them. So it's transmitted to us by a mosquito bite, short incubation period. 20 to 30% of people develop a flu-like illness called West Nile virus fever. But 80% of people have no symptoms. So symptomatically, you wouldn't really know that you had West Nile. You may have in the summertime when mosquitoes are biting a fever and get better, and that would be it. And maybe years later, you participate in a serological survey and they find that you have antibodies, but you were never symptomatic. But the problem here is that 1 in 150 people, the virus gets into the CNS, and that causes problems. Headache, eye problems, muscle weakness, cognitive impairment, sometimes paralysis. There is mortality associated with it, and half of these people never get better. They have long-term neurological sequelae. So we don't have a human vaccine for West Nile virus, but they're working on it because we have a lot of mosquitoes in the U.S. that now carry it because it wasn't here before, but now thanks to an Israeli goose, we have it, and we need to do something about it. So the way that pathogenesis works, right, a mosquito feeds on a bird, say, the birds are infected. Wild birds are all rather infected. But in the introduction in 1999 into New York, many exotic birds in the Bronx Zoo were dying, as well as crows, because they'd never seen the virus before. And whereas birds in other countries had seen the virus for years, they had immunity and so forth. And so it took a while here in the U.S. In fact, first few years, the crows and the blue jays were all wiped out. They only made a comeback later. Anyway, the virus then bites humans. Mosquitoes take blood meals. The virus gets into the blood. It's taken up by Langerhans dendritic cells, brought to the lymph node for sampling to see if it's foreign, of course. But then in the lymph node, there are lots of lymphocytes where the virus prefers to reproduce in. So high reproduction in the lymph node, viremia, which brings the virus to other lymphoid organs like the spleen where it can reproduce even more. And of course, there are innate responses that lead to the flu-like illnesses. Now, remember the other day I told you that the virus is sensed by toll-like receptor 3. That leads to the production of TNF-alpha, which loosens up the blood-brain barrier. And in those people that get neurological sequelae, the virus gets into the brain, can reproduce in neurons. And, eventually it could be cleared by T cells coming into the brain from the periphery. But there's often collateral damage as a consequence. So in the U.S., we have periodic outbreaks of West Nile, so very low, of course, in 99, but then large numbers of cases, 4,000, 10,000 in 2003, and it really depends on the year, the number of cases. I only have data to 2019 here, but they're broken down. You can see neuroinvasive here in 2019, 570 cases, the rest non-neuroinvasive, and 46 deaths. So if you were prone to being bitten by mosquitoes, this could be of consequence to you. This is the incidence by state. The darker states have the highest incidences. We don't have a whole lot here on the eastern seaboard, mostly southern and Midwest. But this is a seasonal disease. Here's the number of cases in 99 to 2019 period by week of onset. So you can see the summer months. It peaks in August, September, the mosquito biting months, and then virtually nothing in the winter as the mosquitoes winter over. And the neuroinvasive disease increases with age. So this is the incidence of neuroinvasive disease per 100,000 population. And you can see with increasing age, the incidence increases. So it probably is related to a reduced ability to clear infection, maybe higher titers, which lead to CNS invasion. So how do you prevent this? Before we have a vaccine, and I suspect someday we will have a vaccine. So if you're, you like to be outdoors, in particular, these culisine species bite from evening to morning, right? So if you're just a day tripper, you don't need to worry. But if you're out evening to morning, if you're sitting on your porch, you should be careful. You should use repellents or screens or clothing before we have a vaccine. There are human vaccines in development, and as I said, there are horse vaccines available because it is 30% fatality in horses. And one of them is a DNA-based vaccine for horses, in fact. A few words on viruses in the CNS. We've talked a lot about this, and this is a topic I've thought about for 40 years because I've worked on poliovirus. So we talked about several viruses today that invade the CNS. Seem to be rare, but they do invade. Poliovirus, measles virus, West Nile virus, they all invade. But remember, they are really transmitted by shedding elsewhere, not from the CNS. So poliovirus is transmitted from the gut, and measles virus is transmitted through the respiratory tract. West Nile, of course, by mosquitoes. So the CNS is a diversion. It is not how those viruses are transmitted. Once the virus gets into the CNS, that's it. It doesn't go back out again. So it's a dead end, and evolution does not select for it. The invasion by these viruses is accidental as a consequence of receptors that are being bound and immune responses and levels of virus in the blood and so forth. And finally, let's say a few things about COVID. COVID is an acute infection. There's no question about it. Here's again Daniel Griffin's slide where we have the time of exposure, and the blue line is PCR detection. We have the time of detectable viral replication by infectivity. We have this is PCR, but detectable viral replication by PCR. Then we have the infectivity level by measuring infectious virus. We have the day of symptom onset, and then a viral phase, during which time the symptom onset is at the peak or just after the peak of RNA, and then it declines rapidly. And so this is basically a 2 to 3 week illness. Eventually, the viral RNA is gone, although it may take a few more weeks for all PCR positivity to go, but there's no more infectivity after that second week or so. And you see the antibodies rise very late in infection, so have little impact on the resolution. So that's an acute infection. But we do have this complication of the inflammatory period, right? A number of patients go into an extended disease where other organs have symptoms. There's overproduction of cytokines. It's an inflammatory disease. And you can see the viral levels are very low. So the virus is no longer driving this. It's an overexuberant immune response that we have learned to tame in some ways by steroids mainly, but we're not yet really great at that. And many of these patients can recover, but all of the patients, whether having mild or more severe disease, are at risk for long COVID. That is a continuation of symptoms of various sorts, whether it's simply loss of smell and taste, respiratory symptoms, lingering inability to breathe and need oxygen for many months, gastrointestinal symptoms, skin problems, all of which really do not depend on virus reproduction. So they are post-acute sequelae, just like post-polio is a post-acute sequelae. So I would say this is, in most cases, an acute infection with long-term sequelae. But in fact, with most viruses, you can find long-term sequelae. With measles, you may have long-term sequelae after encephalitis. The same with West Nile virus. The same with post-polio. So these are all acute infections that have long-term consequences, but that should not surprise us because the viruses are parasites. They're damaging us, and more importantly, our immune response is damaging us as well. And this is just a reminder of the different phases of this acute infection. This is a nice diagram from a review article. Again, asymptomatic and mild disease, 80% of cases. The infection is resolved, the virus is gone. That's the definition of an acute infection. And then more severe disease with increasing age, which is also similar for many of the other viruses we've talked about. Now, after the Thanksgiving holiday, we will return on Monday and we'll talk about the other class of infections that we've referred to briefly at the beginning. We'll talk about persistent infections. And let's go back to some questions. It is very tempting for me to look at I have a separate monitor here to my right, and I see your chat scrolling by. It's very tempting to chat with you all, but I do need to give you the information. And most of the comments are about me not putting the slides on, so sorry about that. And yes, I was wrong about German measles, so that's corrected. This is the best. If I had taken virology before, I would have been a virologist. So thank you. That's probably the best thing you could say, is that I'm influencing you. Yeah. Slides, please. Slides, please. I know. I remember when polio was a highly feared disease. Yeah, it was. And people got vaccinated. And we will talk more about that in the vaccines lecture because there is a twist to that story that we have to tell. Yep. So a lot of people with my slides, but I did repeat everything. Yep. And some of you said, I need to give you my moderators my text because I do have my phone here, and I should give you my text so that you could text me. Yeah, I didn't get it. All right, I'm going to share my text with you guys, and that'll be the solution to the problem because it'll buzz right next to me, okay? Is that good? But yeah, you could go through the PDFs, but you don't see my my pointer, right? And some people like the no slide version. But I'll give you my text, yeah. Okay. Now I understand how I got infected with measles in high school. I walked home next to a friend who was recovering. Yep. He or she was still shedding, yeah. Oh, so that's very interesting. A number of people have recommended Nemesis to me. I would love to read it. I have not. I should. I don't do a lot of reading, although I'd like to. I just brought a new book to the incubator on Tuesday. I brought it from so I have hundreds of books in my office at Columbia. And this is one by Erling Norby, who I interviewed a couple of times on TWIV. And he has insight into the Nobel Prize. Anyway, I brought it on the subway. I read 5 pages on the subway, and now it's here. And I don't know when I'll get to it because I have too many other things to do. It's fine. I'm not complaining. On your study in polio, did you get the best in vitro study that gives you results in vivo? I think the best is that putting the receptor on mouse cells in culture makes them infectable, susceptible, and the same happens in mice. And we know that the receptor governs infectivity in people as well. So Arnold just gets the shivers when I mentioned we infected mice and they got paralyzed, and here's the spinal cord. This is what we have to do. Just like doctors have to deal with death, right? And I'm telling you what I do as part of my discussion, and I'm sorry if you don't like it, but some people have to do all sorts of different things, right? I twice a week go up to Columbia and work with mice. I do all kinds of things with them, and I'm not going to tell you, but if you want information, it needs to be done. If you want to take care of patients, you have to do other things as well. My father was a surgeon, saw lots of people die. Mostly he didn't talk about it, right? No, norovirus does not only infect humans. It does. There have been reports that it can infect other animals, non-human animals as well. We seem to be the major reservoir, so we're spilling back into other animals. But whether it's establishing a reservoir in them isn't clear. That's work by Marianne Koopmans, actually. And she says in one of her papers, we need to monitor it to see what's going on. Is COVID traveling on bacteria? Some people have looked at this. I'm not sure that it tells us anything. So I would say no. Nope. Yes. So some individuals are not cannot be infected with norovirus, as Andrew says here. Yep. Thank you, Holm, for your contribution. Thank you very much. I'm glad you like my style. If you didn't, you wouldn't be here, right? I just love this stuff, and that's why I'm enthusiastic. Norovirus is called winter. I'm not going to try because Kathy Spindler would get mad at me. Winter throwing up disease, yeah. So here in the U.S., it was called winter vomiting disease, yeah. Yes, absolutely. It's worth looking into the microbiome, but that could be protective, and people are certainly doing that. Yeah, Vanity. In the old days, I guess they used, at least in New Zealand, maybe the attendants are very nice and they clean up for their passengers. But yeah, you should not. You should let people clean up their own mess. Yeah. Yep. Jenny uses food-grade hydrogen peroxide. That is actually a great disinfectant, yeah. What about flooding that contaminates ground well water with animal fecal pathogens? Yes. So that's often how cow E. coli gets into vegetables and so forth by contaminated groundwater, yeah. I mean, the number of cows and pigs we raise and chickens is huge, and we really need to get away from that. We need to find other sources of proteins because they cause issues, maybe an influenza, highly pathogenic E. coli, and so forth. And I think, be careful about your eating, right? So Christina doesn't do buffets, Subway, or cafeteria. Yeah, I mean, these mass places where people prepare food for you, it's tough. You have to be careful because even one that's been around for years, there are outbreaks all the time of unwashed or contaminated peppers or lettuce or whatever. It happens all the time. And yes, West Nile virus can cause encephalitis, and you saw the frequency. It is quite high. So I don't know how West Nile got to the U.S. So yeah, some planes get sprayed, but I don't know. I've seen flies buzz out of planes at the end of a trip, so one could escape. I don't think it was a human because humans don't have high enough viremia for a mosquito in New York to have bitten them and transmitted. Or maybe there was one exception. Who knows? Maybe there was a high viremic patient. Who knows? Apparently, the dendritic cell infection, doesn't cause an issue as it does with other viruses. That's right. Thank you, Tom, for your contribution. Really appreciate it. Glad you like learning. Thank you, Barb Mack, for your contribution. What is the blood-brain barrier? Oh, well, we talked about that in a previous session. We had a picture of it. Yes, it's very tight. The endothelial cells are very tight. Plus, there's an astrocyte wrapped around it, so it's very hard for viruses to get through it, or other things as well. How's latency ruled out? You can't rule it out that it's in your... Well, Ebola turned out to be latent in testes. It can last many years there. So, yeah, it could be, but I think I don't see what reason. All right, so with Ebola virus, it's not actually reproducing in the testes. It's just hanging out there, and it's amazing to me that it can last for a couple of years. So maybe SARS-CoV-2 could as well, but I see no evidence for that right now. All the long-term illness doesn't need to have a virus around. Doesn't mean it's not there, but I don't think you need to invoke that unless we have data. Thank you, John, for your contribution. Really appreciate it. And many people, what's more effective against COVID, testing or vaccinating? If you could test everyone every day, that would work, but we can't, so we have to vaccinate. We do not know why people develop a hyperimmune response. We think it may be in part because of a poor innate response initially, and that lets viral loads get very high, but not clear yet. We'll figure it out. We're working on it. Sequelae. I'm glad you learned a new word. Isn't that a good word? I like sequelae also. Yeah. And we have a link for you to look at in Twitter, so check that out when you have a chance. You have an iPhone SE. Yeah, this is an SE. I bought the red one. Why did I buy this? Because it's the cheapest one. And I don't use it that much, but I do have a case with my virus with polio. Unfortunately, it's got vaccine stickers on it, so I have to get a new case because I don't want to throw those away. Let's see. I'm going to wrap through these now. I know it's a bit late, but did polio damage neurons? No, it seems to be strictly motor neurons, interestingly. Never any sensory alterations in polio, neurologically invasive polio. It's very interesting. Is there any reasons that damaged neurons cannot repair? No, they don't. Neurons do not repair, especially the motor neurons in the cord. That's it. That's the end. There's a typo in the norovirus. I think someone else pointed that out as well. Let me see. Where's the typo? Vomiting. Maybe it's my typo. All right, I'll have to look into it. I don't find it. The second half is slightly easier than the first. Yes. This pathogenesis is more descriptive than the first half, which was highly molecular and depends on more background. Yeah. Yes. Erling Norby, wonderful guy. Just listen to the TWIV. There were two. And he's from Sweden, and I would really love to go back and visit him. Maybe next year a trip to Sweden and maybe Finland and Norway is in order. Visit some big virology centers and do some TWIVs. Aren't viruses a sort of law of nature to keep the number of species within limits? I don't think they're a law, but I think they do that, like parasites. Dixon say parasites keep species numbers to a certain level. I don't think that was the original intention because they evolved on their own, but that's certainly one of the outcomes, yeah. Thank you, Thomas, for your contribution. Really appreciate it. And, oh, yes, tonight we have a Q&A, right, at 8 p.m. Eastern. More questions, you can bring them there. The two of us are happy to chat. I hear a lot of people say being cold makes them sick or that change in the weather. Yeah, I think those are associations. It's like I tied my shoe and it rained. Is that causative? No, it's just an association. So I think there's myths. Yeah. Okay. Let's see. That will do it. That's all for the questions for today. Thanks everybody for coming here. 128 of you, pretty darn good for the day before Thanksgiving. It's better than some of my classes have been. I've had one person show up on the day before Thanksgiving. So happy Thanksgiving, everybody. Be safe, be smart, and we'll see you next week or tonight at the Q&A. If not, we'll see you next week. Take care, folks. Bye-bye.